Bismillahirrahmanirrahim. My name is Sadia Bibi. I am going to present the topic is regulation of gene expressions. Introduction of regulation of gene expression. What is gene? Gene is the basic unit of heredity. And what is regulation of genetic expression? जब किसी जीन की इंफॉर्मेशन से कोई न्यू फंक्शनल प्रोडक्ट बनी होती है तो उसको कहते हैं रेगुलेशन ऑफ जीन एक्सप्रेशन. सो रेगुलेशन ऑफ जीन एक्सप्रेशन इंक्लूड अ वाइड रेंज ऑफ mechanisms that are used by a cell to decrease and increase the production of a specific gene product, protein or RNA. Sophisticated programs for gene expressions are widely observed in biology. For example, to trigger developmental pathway response to environmental stimuli and adopt to new food resources. So regulation of gene expression जो थे ना also used in development, growth, response to environmental stimuli के लिए और food resources के लिए. Next, virtually any step of gene expression can be modulated from transcription initiation to RNA processing and to post-translational modification of a protein. So gene expression जो होते हैं ना उसको वो transcription initiation, RNA processing और post-translational modification of protein के लिए भी use होते हैं. Often one gene regulator control an other and so on in gene regulatory networks. Gene regulation is also essential for viruses, prokaryotes and eukaryotes as it necessitates its versatility and adaptability of an organism by allowing the cell to express protein when needed. Gene regulation जो होती है ना यूकैरियोट में और प्रोकैरियोट दोनों में होती है. The process of alteration of gene expression has been studied in detail in prokaryotes. It generally involves the interaction of specific binding protein with various regions of DNA in the immediate vicinity of transcription sites, and this produces either positive or negative effect on transcription. So, in eukaryotes, studies are not extensive and not well understood. प्रोकैरियोट के अंदर इसमें जिसकी जीन की एक्सप्रेशन को हम डिटेल से स्टडी कर सकते हैं, लेकिन यूकैरियोट के अंदर इसका जीनोम जो होता है, इसका जीनोमिक स्टडी जो होती है ना वो एक्सटेंसिवली हाई होती है, तो उसके लिए इसलिए हम इसको यूकैरियोट में नॉट वेल अंडरस्टूड नहीं कर सकते. सेल इन यूकैरियोटिक यूटिलाइज द बेसिक सिंपल प्रिंसिपल बट यूज अदर मैकेनिज्म टू रेगुलेट ट्रांसक्रिप्शन. नेक्स्ट, type of gene regulation. Mainly two type of gene regulation: positive regulation and negative regulations. Positive regulation, when the expression of genetic information is quantitatively increased by the presence of specific regulatory elements, it is called positive regulation. The element or molecule mediated positively regulation is called positive regulators. Next, negative regulations, when the expression of genetic information is quantitatively decreased by the presence of specific regulator element, it is called negative regulation. So negative regulation में जब जीन एक्सप्रेशन की फ्रीक्वेंसी रेट कम होगा तो उसको हम कहेंगे नेगेटिव रेगुलेशन. सो द एलिमेंट और मॉलिक्यूल मेडिएटेड पॉजिटिवली रेगुलेशन इज कॉल्ड नेगेटिव रेगुलेटर्स. आगे, टाइप ऑफ जीन रेगुलेशन ऑन द बेसिस ऑफ द फंक्शन. टू टाइप: इंड्यूसेबल जीन एंड कॉन्स्टिट्यूटिव जीन. Inducible gene, the expressions of inducible gene increase in response to an inducers. What is inducer? Inducers that are used for transcription and are also used for enhance transcription. Inducer are small molecules. Some protein produced by Escherichia coli. Escherichia coli जो होते हैं ना present होते हैं बैक्टीरिया के अंदर. So beta galactosides are said to be inducible because they are not only produced in significant amount but also but when a specific induced lactose is present, lactose जो तो present होता है लैकोप्रेन के अंदर. आगे है tryptophan pyrrole of liver is also induced by tryptophan. डायग्राम इंड्यूसर जो प्रोमोटर्स होते हैं, इसके अंदर 3 टाइप ऑफ जीन्स होती हैं. इससे आल्सो कॉल्ड स्ट्रक्चरल जीन्स. और इंड्यूसर्स होते हैं रिप्रेसर होते हैं. रिप्रेसर जो है उनका मेन फंक्शन होता है जीन को रेगुलेट करना. नेक्स्ट, constitutive gene. The constitutive gene are expressed at the more or less constant rate in almost all cells and they are not subject to regulations. The product of these genes are required in all the time of cell. For example, enzyme of citric cycle. Next one cistron and one subunit. One cistron, one subunit concept. Earlier hypothesis proposed that one gene produced one enzyme or protein, and one gene one enzyme concept was introduced. So it is also known that some gene and protein molecule are composed of two or more non-identical subunit which can be explained by one gene and one enzyme theory, so it is not valid. The cistron is not considered as a genetic unit coding for the structures of the subunit of an enzyme protein molecule acting as a smallest unit of genetic expression. Cistron जो होता है ये एक genetic unit होता है जिसके जो तमाम जो होती है ना genetic information को express कर रहा होता है. Here one gene one enzyme idea might be accurately regarded as one cistron and one subunit. Next, gene expression in prokaryote operion. The concept of operion was first introduced by Jeff Bone and Modon in 1965. Lac operion जो होता है ना it required for transport and metabolism for lactose in Escherichia coli. ये उस वक्त required होता है जब transport करनी होती है और metabolism करना होता है lactose का in Escherichia coli के अंदर. operion जो होता है it is also structure of DNA. operion it is also called genes. So operion is defined as a segment of the DNA strand consisting of structural genes, operator gene and regulator genes. So first of all, structural gene are cluster of strival structural gene which carry the codon which can be translated into proteins. So next is operator genes. One operator gene which has overall control on the process of translations. Operator gene के जो control कर सकता तमाम process of translation को. Next one is regulator genes. Third gene is called regulator gene. It is also located sometime at the distant from the operator gene on the same DNA strand. Regulator of gene transcribe the messenger RNA which are synthesized the repressor proteins molecule which regulate the transcription. Transcription क्या होता है DNA से messenger RNA का बना it's called transcription और messenger RNA से protein की synthesis is also known as translations and receptors are also used for regulator the genes. Next one P site. P is associated between operator genes and regulator genes. Next is the lac operion is also inducible cataboloperion of E. coli, Escherichia coli. It is also consists of structural gene. It carries three structural genes Y, Z, and A, code respectively for beta galactosides, beta galactosidase, galactoside pyrimase, thigh galactoside, and transacetylase. Function. Beta galactosidase hydrolyze lactose to galactose and glucose. जो होता है बीटा galactoside जो होता है वाटर की मौजूदगी में लैक्टोज को कन्वर्ट करता है ग्लैक्टोज में और ग्लूकोज में. सो ग्लूकोज होती है ग्लूकोज जब लैक्टोज ज़्यादा और ग्लूकोज कम हो तो लैक ऑपरियन के अंदर तो जीन की रेगुलेट होता है तो जीन रेगुलेट हो जाता है. नेक्स्ट वन इज़ primase is also responsible for the transport the lactose into the cells and acetylase coded by A gene is not known properly. Primase जो होता है ना that allow the lactose to enter the cells के अंदर enter होता है लेकिन acetylase का कोई proper function नहीं आता. Structural gene YZA transcribed to form the single large messenger RNA with three independent translation unit for synthesis of three distinct enzymes. Such a messenger RNA coded for more than one protein. It is also known as polycistronic messenger RNA. The वो messenger RNA जो कोड कर सकते one से ज़्यादा protein को it is also known as polycistronic messenger RNA. Next is next one. Its structure of lac operion. I also represented the regulator gene जो regulator gene को करती है इसके आगे अगर promoter side P or also operator Z Y A. It is also the genes of lac operion. It is also called structural genes. Next is it is also structure of lac operion. इसमें promoter of the structural genes Z Y and A. Z is also known as beta galactoside. Y is also primase and A gene is also transacetylase. जब बीटा ग्लैक्टोसाइडेज होता है वो लैक्टोज को कन्वर्ट करता है ग्लूकोज और ग्लैक्टोज को फिर ये आगे कन्वर्ट होता है कार्बन डाइऑक्साइड वाटर और एटीपी के मॉलिक्यूल में. जो प्रीमेज होता है जो वो डेट अलाउ द लैक्टोज इंटर इनटू द सेल्स एंड एसिटाइलेज का कोई प्रॉपर फंक्शन नहीं होता. Role of catabolic gene activator proteins. Catabolic gene activator protein is a dimer that acts as a positive regulator of many catabolic operon like glycoprotein in Escherichia coli. Attachment of RNA polymerase to the promoter site in the presence of catabolic gene activator bound to cyclic AMP. Absence of the glucose in cell activate adenylate cyclase, which catalyze the synthesis of cyclic AMP from ATP. The later bind of CAP to form a CAP CAM complex. Unlike the free CAMP, this complex binds the promoter site immediately. The stimulation initiates the transcriptions of lac operon structural genes in the absence of repressors. The bind of CAP and cyclic AMP complex to promoter enhance the RNA polymerase activity. On the replacement of the glucose, lower the intracellular CAMP. जो ग्लूकोज होता है इसका भी फंक्शन होता है लैकोप्रियन के अंदर. सो ड्यू टू डिक्रीज द लेवल ऑफ CAMP द फॉर्मेशन ऑफ CAP एंड cyclic AMP complex to the P site is negligible and transcription does not occur. The glucose interferes with the expression of glycoprotein by depleting the CAMP levels. Gene expression in eukaryote. Gene expression in eukaryote can be same as that in prokaryote because of cannot say genome of eukaryotic is much larger than prokaryote. Genome is a genetic material का structure होता है वो eukaryote में बड़ा होता है than prokaryote की निस्बत. Human genome also consists of 32 pairs of diploid chromosome होते हैं that also contain 109 billion base pair of DNA with the estimated 1 lakh gene approximately. There is a great deal of redundancy in the DNA of eukaryotes. Prokaryotes, like the repressor protein, influence the gene expression at the DNA level as they synthesize outside nucleus and cannot get into nucleus through the nuclear membrane. Thank you.